Hello, my name is Yasuyuki Tanahashi. I'm an associate professor in the Department of Animal Medical Sciences at Kyoto Sang University. Today, on behalf of my courses, I'm very happy to talk to you about the key results from a published paper.
In the small intestine, stimulation of M2 and M3 muscarinic receptors expressed in smooth muscles activate cationic channels, leading to membrane depolarization. This depolarization opens voltage-gated calcium channels, resulting in muscle contractions. ATP-sensitive potassium channels, referred to as potassium ATP channels, which is a keyword in our paper, are well known to be expressed in gastrointestinal smooth muscles. These channels have been reported to be inhibited by muscarinic receptors. However, much remains unclear regarding the underlying mechanisms, such as which subtype is involved in this suppression. To shed some light on this point, we used M2 or M3 muscarinic receptor subtype knockout mice. We also investigate the physiological roles of the muscarinic suppression of potassium ATP channels in the regulation of ileal muscle contractility. First, we investigated the effect of the potassium ATP channel blocker glibenclamide on membrane potential with patch clamping, and on contractility by tension recording in mouse ileal smooth muscles. After application of glibenclamide onto a single smooth muscle cell isolated from mouse, membrane depolarization and firing action potentials occurred. Glibenclamide induced contractions in a mouse ileal segment. These results suggested that potassium ATP channels are constitutively active and contribute to the setting of the membrane potential and regulating muscle contractility in ileal smooth muscles. Next, to elucidate the mechanisms underlying muscarinic suppression of potassium ATP channels in ileal smooth muscles, we investigated the effect of the muscarinic receptor agonist carbachol on potassium ATP channel activity in smooth muscle cells isolated from mouse small intestine with patch clamping. This represents raw data. After potassium ATP channel current induced by the channel opener chromacalin reached the plateau level, additional carbachol was applied. As you can see, the amplitude of this current was strongly suppressed by carbachol in wild-type mouse cells. Furthermore, we performed the same experiments in cells isolated from M2 or M3 receptor subtype knockout mice. As you can see, carbachol induced comparable suppression in M2 knockout cells to wild-type cells, but minimal suppression in M3 knockout cells. In conclusion, M3 receptors can inhibit potassium ATP channel activity, contributing to membrane depolarization and resulting in muscle contraction.
Our results provide basic information on the muscarinic regulation of gastrointestinal motility, which may help in developing new medicines to treat function bowel disorders, including irritable bowel syndrome. If you have any questions or would like to read our paper, please do not hesitate to contact me. Thank you so much for your attention.